Okay, so let us look at this microarray. So what's microarray? Is something like a computer chips. Okay, so microarray, if you look at the entire things, it's very, very small. The other chip, can you see that? It's about 1.28 centimeter by 1.28 centimeter. Can contain thousands of the well inside. Can you see that? So very small, but we can actually have it's a high throughput screening. Basically means that you don't screen for one or two genes, you screen for thousands of the gene at the same time. Okay, so as the genome and number of species have been sequenced, now it's possible for us to study the expression of large group of the gene. So researchers use what we know at the whole genome to investigate which gene are transcribed in different tissue or different stages of development. Let's say from fetus to embryo to fetus to the offspring. Can you see that? Okay, so we can use this what we call the DNA microarray assay. So what's a bit special is something like DNA chips. What we call a DNA chip is because we can actually read it and using computer or software to help us to analyze it. Okay, so a DNA microarray actually consists of single-stranded DNA fragment representing different genes fixed to a glass slide in a tightly spaced array or a grid. So you can see that this is a different well. Different well actually have the different single-stranded DNA fragments which represent a particular gene. Okay, I will show you guys later. So microarray can be used to detect which gene being expressed at any specific time in each cells in the body. For example, if I want to look at expression of the cancer cells are different from those active in the non-cancerous cells. Okay, so let us look at what is this microarray actually. If you look at a microarray, this microarray actually, they have a grid. Something like a well. So if you enlarge each well and view it from the side, you can see something like this. This is a well. Okay, for let's say this is a gene number 1. Okay, gene number 1. So because we already know the sequence of the genes already, so what we're going to do here is we embed or we call this we conjugate, okay, or immobilize the single-stranded DNA that corresponding to the gene 1. So what's mean this mean? If gene 1 is A, T, A, C, G, G, C, C, G, G, C, T, A, for example. Okay, so this is the gene. What we're going to do here is we synthesize the single-stranded DNA, which is complementary to the gene. Are you clear? We synthesize it complementary to the gene. So it means that we're going to have what? We're going to have, okay, let's say I detect this region. Not all, I can detect a short fragments enough already. So it means that here will be TAT. Okay, I have T, sorry, from the bottom. Okay, let's say TAT. Okay, and then GCCGG. So GCCGG. Okay, so represents gene 1. So I have the gene 2, gene 3, gene 4, gene 5, and so on. Okay, this means that each gene, okay, each well represent one gene. So we can see a single-stranded DNA. Okay, understand? So this is microarray. So each well represent a gene. Why? Because we embed the single-stranded, okay, we embed or immobilize the single-stranded DNA that represent the particular gene in this microarray. So means that we can embed thousands of the gene, okay, in one microarray. Okay, so understand the microarray. So what will happen here is, let's say we want to study cancer cell. Let's say breast cancer. Okay, so we take the breast cancer cell. Okay, and then we take a normal cell, normal breast cells. Okay, now we want to study what kind of gene actually causes breast cancer. What kind of gene actually they do not have any activity on the cancer cells. So how we can do this? How we can do this? Basically very simple. what we're going to do here is we extract the mRNA from both samples. Okay, we extract both sample mRNA. So what we're going to do here is we're going to use a reverse transcriptase. We use a reverse transcriptase to onto both sample. Okay, but special this time. The nucleotides that we use here, they are fluorescence labeled. Let's say for the cancer cells, we label it with the red fluorescent dye. So means that if I use a reverse transcriptase, this is mRNA. mRNA will be used as a template, or mRNA will use a template to form the cDNA, the hybrid, and then we degrade mRNA, make all these cDNA, they are stained or they are labeled with the red fluorescent. So this is cDNA. Clear enough? Okay, still remember we use the reverse transcriptase, okay, to get the cDNA, and this cDNA now they are labeled with the red fluorescent dye. Okay, same goes to this mRNA from the normal, okay, from the normal individual or normal cells. Then we use the reverse transcriptase to form the cDNA. Clear enough? So this cDNA now they are labeled with a green fluorescent dye. So this cDNA, the total mRNA. Are you clear? All mRNA, I don't care. Okay? All mRNA, then we're going to get this. Okay, we get the cDNA already. Okay, done. Now, what I'm going to do here is very simple. We mix both samples together. We can differentiate them, eh? Can, because one is blue, red color, one is green color. Then we load them into the microarray. We load into the microarray. So in the microarray, what will happen here is because cDNA is double-stranded, right? So we have to denature the cDNA, and then we let the hybridization take place. Now, what is hybridization? In this process, the single-stranded cDNA, it will hybridize to the single-stranded DNA probe, that immobilized one. Are you clear? It will immobilize it. So what will happen next? We're going to wash a few times with the buffer, and we can read it by using the microarray reader. Then we can interpret the result. Okay, so I will teach you guys later how to interpret the result. But now we look at the principle. What actually happens now? If I say I have 5 wells, well number 1, well number 2, well number 3, well number 4. Okay, 4 wells, sorry, 4 wells. Okay. Now, each of them, they have the probes. Black color probes, different probe. Okay. Now, what will happen here is, a total cDNA, total cDNA. So you have the red one, you have the green one. So which one can bind depend on the gene expression. If gene does not, the particular gene does not express, means what? They don't have the particular chrono. So, okay. So what will happen here is at the gene number 1. Let's say the red color bind, hybridize, 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 hybridize. So the extra one, we wash it away. Okay. Let's say gene number 2. Okay, so total DNA got a red color, a cDNA, red color and green color. Okay, but only green color bind. Why? Because the cancer cell didn't express chrono. Okay. So the third well also the same. We have the green color and red color, but now you can see both of them bind because both of them also express that particular gene. Okay, Then we wash away that unbound. And then the last one also, you have the red color and the green color, but none of them bind. Why none of them bind? Because both of them didn't express. This is the, if you look at this one, they don't have a sequence that complementary. So that's why they cannot bind. This is total. Can you see that? So those unbound one will be removed. Gene number 3, gene number 4. So from here, then we do know that, okay, if I read it, so this one will give me the red signal if exposed to the UV. This one will give me green. Then how about this? It will give us the yellow, yellow-orange color. And the last one, you won't have any signal, no signal. So it means that you're going to have 4 different kinds of the result. So if the red color, like in the, number 1, gene number 1, because it gives us a red fluorescent signal, so indicate that cancer cell is the one that express. Normal cells, because no green color, right? No express. So it means that this gene might be the one that causes cancer. Can you see that? Only expressed in the cancer cells, but not in the normal cells. So it means that most likely this gene is expressed. Okay? Now, number 2, gene number 2, you only see green color, green fluorescent signal. So it means that normal cell express, cancer cell does not express. Can I see that? So you don't target this gene, because you target this gene using a drug to bind to this enzyme, for example, this protein, it might cause cancer. Are you clear? Clear or not? Okay? Or it will kill the normal cells. Then how about number 3? Number 3 give us orange color or yellowish color. Depend on intensity. So means that normal cell express, cancer cell also express. So means what? It's nothing to do with cancer lah. Correct or not? This gene just normal. It's a normal genes that both cells also express. So it means that you don't going to inhibit this protein. If you inhibit this protein, normal cell may not be able to survive. Are you clear? Okay. So number 4, no signal, no fluorescent signal. So in this case means that normal cell not express, cancer cell also not expressed. So no point actually you're going to study these 2 genes. I clear these 2 genes no need to study because they are not related to the cancer. So you can see that the expressions of the particular genes, we can actually study them based on what? Based on the expressions using the microarray. So if you need, you can actually take a photo for the process first, the microarray. And then no need to copy down. Don't waste don't waste time to copy down. Okay, you can always take the photo and then you can print it out, or you can actually store it. Can you see that's the interpretations of the result? And the color intensity, how strong the intensity also tell us the how strong the intensity also tell us the activity. Higher intensity means that express more. Lower intensity basically means that express less. Okay, so intensity also will give us the clue. Okay, let's say for example, the particular red color, so many of them red color, some of them just express in a small quantity, then we ignore them. We study those that have high intensity. Okay, so this is a microarray analysis. So as I said, that cancer cells, we use the RNA, we isolate the mRNA, use the reverse transcriptase, and with the label, can you see that from cDNA, we form a cDNA, but for cancer cells, it's a red color, normal, green. So when you put it in this, can you see that some of them give me green color, some of them give you a red color, some of them you see no color at all, right? And no signal, some of them give you yellow or orange. Then we have to analyze it later, okay, by using the microarray reader. So you can try to attempt the questions. Here is the answer. It's a PCR also, because we want to form cDNA, okay? So it deals with the PCR. So we need the denature, so they give us single-stranded, then it can hybridize with the probes, okay? So intensity of the light actually indicates the activity, and also we will know which gene actually express, Okay. So how about the data analysis? So researchers synthesized two sets of cDNA, fluorescently labeled red or green from mRNA from cancerous cells and normal cell respectively. So red color for the cancerous, green color for the normal. Okay, understand what it means respectively. So it means that first one match with first one, red or green, cancerous, normal respectively. So it means that red for cancerous, green for normal. Okay, so the results show here. The results show here. So it hybridized with a microarray containing 5,000 over gene. Okay, resulting in the pattern shown here. So the intensity of fluorescence at each point is measured in the relative expression of the two samples of the gene represented by the spot. So you can see that we have red color, green color, orange color, and also no signal. So what is the conclusions here? Conclusions. So first, cancer cells express. Normal cell, no expressions. Green color, cancer cell, no express, but normal cells express. So orange color, cancer cells, yes. Normal cells, so yes. Okay, no color signal means no hybridization, right? No expression. That's why you don't have the mRNA that can complement and as a cDNA that complementary and attach to the probe. So it means that here, cancer, oh, sorry, what happened? Okay, cancer cell, no. Normal cell, no. Okay, so with this, I have done for the microarray as well. Okay, so any more question, guys? Any more question? So you need to know how to explain. First, principle very simple. Okay, why we use the microarray? Microarray is the one that actually help us to detect the expression based on the mRNA. So we can isolate mRNA and then from cDNA, fluorescent label them, then we can load them into the microarray and we see the intensity. Okay, so understand this enough already. So then you need to know how to interpret the results. Okay, so with this, I have done the microarray already. Now you can go to register your attendance. Let me off the record first.